learn a bit more, learn some tools to really work on a data set that I've collected in the previous months. And I'm currently doing a PhD in cognitive neuroscience. I work in neuroscience and music. Basically, my data related to a musical performances, sung performances, improvised sung performances. And this is what typically a sung performance looks like when it is processed with a software that extracts automatically the frequency, the pitch and the frequency, and therefore extracts automatically the notes that are sung. My idea was to learn how to possibly use new analytical techniques to investigate the data that I collected and investigate possible regularities and statistical properties in improvised performances, to possibly also see how people implicitly learn and acquire implicit musical knowledge. So to do that, since I have a huge data set, I need to know how a script, I need to learn how to write a script that goes into folders automatically, loop into folders, extract, identifies and extracts exactly the type of files that I want to, and concatenates them and displays them. So here, since the software that I have initially used, which extracts the information, gives a txt file, I will be using just the txt files, and I will concatenate them in this way. This is something that I've done in few days ago. My idea is to, here you can see the seconds, the frequency of each note, and the duration of each note. And the idea, and this goes on for each performance, and the idea is to have a huge data set here, and then run some analysis on this. And this is what I just took a screenshot of what my dataset looks like. Those are all the participants, and those are all the files that the software generates in the TXT format. So the idea is having something that is able to combine all of this in an organized way and visualize it in a way that makes sense, and being able to work on this. And so this is the type of steps that I have wrote for this course. And the last goal would be to identify a possible analytical technique to investigate what I have. I know that it sounds very general, but it's just the beginning of my project, and I just started working on it. So it's a very exploratory phase. What I have now on my hands. So that's it. Don't be. It's very fancy, as you can see. It's very. Anyway.
So as a bit of context, Emanuele had a health issue last week. He wasn't able to attend week one, So he hasn't necessarily had all the exposure to the catalog of tools that you have had. So do you have ideas of tools he could be using here? Because so far I just mentioned pandas. So do you have suggestions for him in terms of what he could be trying out?
To visualize data that are stored in a pandas data frame, I would suggest you to use Seabound because the links between the two libraries are very fluid.
Okay.
And it's like one line of code to have a pretty nice graph of your data.
Seaborn?
Seabound, yeah.
Okay. Thank you. Thank you.
And yeah, I hope you're like on your GitHub opening an issue right now.
That's a good link of the back. Yeah. Yeah, exactly.
In terms of the exploratory data analysis, what have we seen that actually Seaborn does them? Like when you, if you visualize a correlation matrix, for example, it does a cluster analysis for you behind. Like it's both a visualization and a sort of lightweight analysis library. But what other packages have we seen? For exploratory data analysis. sklearn is a good one. Scikit-learn. Can definitely do a number of things down there. For more statistical analysis. Don't you remember? Well, you have like a gazillion of them, yeah, for sure. Huh? No, you don't remember the stats model? You haven't talked about stats models on week one? Ouch. Okay, so stats models are like most, linear regression, multiple regression, multi-level regression analysis, all in stats models. And I think you've seen also Neo tools, something like that. Bratislav's library. Somebody was telling us about it earlier for PLS analysis.
Net neural tools on Python.
So Neo.
Net neural tools.
Net neural tools.
Net neural tools.
But for the repeated, you might just want to, I don't know, there's probably some mixed effect. For the repeated design might be, so it's that.
So once again, jump, GitHub works well on a cell phone. A quick issue here. Yeah, so there's lots of, I think that's one area where Python really shines, is more like, exploratory data analysis with scikit-learn is first class, but there's a number of alternatives you can try. Pierre?
Do you still have the audio files?
Yep, of course. They are here in each folder. They're here.
I mean, like you got audio files, like the original recordings. The recordings of this, like.
How did I got them?
Do you still have them?
Yes, I still have them.
Because there's a ton of Python libraries dedicated to acoustic, auditory feature extraction, music feature extraction, where you would get like all the amount of different features, spectral lines, temporal lines. It would be nice like to get the whole workflow into that kind of
Yeah,
they have like all of it. You can extract like
the only it's a good suggestion. The only thing is that when we work in this field, there is a lot of work that we do to correct what the software, like in order to identify the nodes. There is a lot of manual work because now the software are able to they come to the point to be able to extract the nodes, which is already a lot, but there is a lot of manual correction to be precise, try to identify as precise as possible what people are singing, what they are producing. So it would be great to know if what you mentioning could give also a way to address this, because
they have like all the algorithms for like automatic segmentation into words,
okay.
nodes, you get a percentage of probability for each node, how much energy is a certain chromogram or frequency band, it's all there.
Okay, nice.
I'm doing issue on that, yes.
Awesome. So maybe you
I already
can,
tried to
you can like meet and, I mean, that wasn't necessarily one of your core objectives, but clearly it would be great to see if you could have like an end-to-end Python workflow
Oh, yes.
from the raw signal to the stats. It'd be wonderful. Okay.
Okay.
Any other suggestions? We're gonna
Thank you.
move on. Thanks a lot. So, Mike.
Hello. So I was. Oh, do you listen? Do you hear? Yeah. So I was pleasantly surprised when I saw that I'm not the only one working with animals. So it's just I'm gonna try to explain you what kind of experiments I do. So I've been jumping between basic and clinical research, but now my PhD is on neuroscience, and I'm working in the auditory cortex of rats and looking at how plasticity is shaped by experience, by age, by drugs, etc. So the data I use in this particular experiment, I have 24 rats, and I train them in an auditory discrimination task. This means that they have to learn to identify a sound. It's a train of sounds, and then one of the sounds is different. If you present the rat with that train of sounds and one is different, the rat has to learn to poke and it will receive food. If all the sounds are the same, the rat won't receive any food. So as the trial progresses, if a rat correctly identifies the sounds, for example, then the next trial or the next time, it will be more and more difficult because the different tone will be closer and closer to the others. So anyways, it's an oddball discrimination task. It's an adaptive task. So that gives you the opportunity not to only look at how fast rats are learning, but also how well they are learning. So this is the kind of behavior that I have. And over a time of 60 sessions, you see how their performance steadily improves. But here's one of the things I'm doing is by doing some treatments. We can discuss later if you are interested. With some genetic modifications, I have 3 groups: a control, a group in which I am inhibiting a particular type of cell in auditory cortex, and another group in which I am exciting those cells. So long story short, we see that in the group that I'm shutting down those specific type of cells, those rats seem to be learning faster. So this is the blue line, and this is the criterion. So I have all this data per session for all these rats. And normally what we do is we look at behavior, and then we do brain recordings. We present a bunch of sounds, a bunch of frequencies at different intensities. And then for each neuron, we can reconstruct, okay, this neuron likes these frequencies at these intensities, and then we call this a receptive field. So we have this electrophysiological data, and we also look at the correlation between neurons. So it will be, it's not the same as resting state, but you are recording at the same time from 60-something neurons, and then you look at how well their signals in silence, just in a resting state, how well they correlate. So normally what we do is we report the behavioral data, we report the change in receptive fields, and we report separately the correlation or the cross correlations. But what I would like to do is to integrate these awesome techniques I'm learning from to actually try and see if there's a way to extract from the behavioral data to pick up what are the features that are more predictive of the changes I expect to see in the brain recordings. So you will be surprised, but that's usually not done in the animal world. So that's the challenge I want to work on during the BrainHack. So after talking a little bit with Jake and after seeing Bratislav's presentation, I will try out this PLS analysis, the partial least squares, and I'm gonna then try to cross-validate. And it's to be defined, but I think I might be using leave one out approach. I would appreciate your comments. I need to read more to see if that will be the best way of doing it. So my to-do list, because I am Python impaired, I just have to learn to just transfer my data from MATLAB, try to work and make these libraries, try to find out which is the right toolkit. So this is what I'm doing. Actually, I'm finalizing my if-is analysis on MATLAB, and then I will translate all that to Python, and yeah, we'll see what happens. It's more of an exploratory analysis, but just to be fair, I would like to first try with the variables that I think will be the most important, and then do exploratory analysis to see what kind of features I might have missed and I can pick up. So yeah, thank you.
In terms of your deliverables, like what would you want to sort of generate out of this? So a script to do those analyses. That would be your end result.
Yeah, but not the script, the actual the result of the PLS analysis. So I would like to actually come up with an answer, albeit simple, like, okay, which are the behavioral features that will better predict the changes in brain connectivity and in brain properties.
Okay.
Yeah.
So you're after the science.
What?
So your outcome is the science.
Yes.
Okay. It'd be great if you can manage to put that to those notebook thingy.
Mm-hmm.
They will give you the figure and you can really put text in it.
Okay.
So you can both do a summary of your findings and a code. It's kind of like what the notebook format is about.
Yeah. Okay.
So I mean, a lot of people have said that would be their outcome, and I think here that would be also very adapted.
Okay, perfect. Yeah, so it was not an original objective, but yeah, I can surely try if it works, and to have an output as a Jupyter notebook, for example. Yep. Okay.
And are those data open, or they're?
Yeah, those are my data, and I can make it public. I made the mistake of putting it directly on GitHub, but then like Jake opened an issue, and they're like, okay, no. So I'm gonna try a better repository, and then I'm gonna link from my GitHub to that.
Okay. So there's like a couple of really like interesting points out of this. Git is very bad at handling data. When a repo becomes big or has many files, it sort of crumbles under its own weight. Typically, if you add data in your GitHub repo by mistake and you remove it, the data is in your GitHub repo, because GitHub keeps track of every changes you ever make. So removing something from a repo, you can always check out the version where it was there, it will be back there. You can actually do surgery on your GitHub history logs if you want to actually remove something from your GitHub repo. You know, like, good to know. And nobody has put as outcome actually releasing a data set. Properly releasing a data set with proper documentation on a portal such as Figshare or Zenodo. It's itself like it's a big job. And it's a very interesting process. So if you actually are okay with putting that data online, and that's something you would see value in, then you could definitely put that as one of your deliverables. And you're probably gonna have a DOI for your data set. Some people may actually start using it. So that's cool. And also, cleaning the data, visualizing the data is, I mean, the sort of, you have all those memes going online that says that data science is 80% data cleaning and 20% data modeling. And it's also a big job, and Panda is wonderful to do that. It's got like all those really neat little tools to visualize the data. I think, like, I haven't had a chance of trying it out, but there's this report for data quality that is all the missing values, give you correlation between your different variables, things like that. So that could be really an outcome of that project. Just try to sort of like QC your data through pandas. It would be a wonderful exercise, I think.
I think because of the time constraint, I will bypassing that, because I already did that on MATLAB.
Already
so,
done that in MATLAB.
So
you know that it's good.
But then it will be it will be another new project to compare the outcome of MATLAB cleanup versus Python.
Absolutely. but Pandas is really a cool library for manipulating data, and I've stopped using MATLAB a little while back. But at least when I was very familiar with MATLAB, it was nowhere near what you can do with Pandas. So it's really worth checking out if you have the time.
Thank you.
Questions, comments? No, all good. I mean, it's amazing. We now have rat electrophysiological data, so we're covering it all. We've got electrophysiology experts in our BrainHack school sort of network, Andrea Green and Paul Sichek. So if you need support on that front, we could ask them, but I feel like you're on top of this.
I can always learn more.
All right, thanks a lot.
Thank you.
Okay, so far so good regarding time. Well done, everyone. It's not easy to keep on track. So next project up is François. Oh.
So hi, I'm François. I'm working at the Cymex lab. As you can see here, I saw the other François upload this image. I thought it was a good idea. I'm working on the memory task. We basically try to reproduce the CIMAQ data set. Not sure how to say it in English. So basically we show trials of 15 images displayed in either one of 4 quadrants. Images are cropped and resized to be 500 by 500 pixels, and the subject has to remember if a target has been repeated or not. And if so, has to mention where it was seen earlier. Maybe I can find a way to launch my demo. Do you have PsychoPy on your computer?
No.
No, okay, I'm just gonna go straight ahead. So basically this repository has everything you need to kind of understand what we're doing. I posted some issues and I'm asking for your help because there are some minor bugs that are occurring. For example, in the function I made to uniformize the images, I don't use any rotation method, but sometimes some images are rotated for no reason. If someone good at that could help, that would be very appreciated. So basically it's not a big project yet, but I'm working to create the program to launch the monitor which will display the images, find a way to record every as much data as possible from the subjects. And also if you guys ever have an idea to make this task less painful for our subjects, it would be great. We need imagination. It's hard to achieve the same reproducibility without by changing it, but if you ever have a good idea, it would be perfect, because some subjects have reportedly fallen asleep, so we need to increase
memory encoding task.
Memory encoding task.
So you're showing them images and they need to remember them.
Yeah.
Is correct? And people were falling asleep while doing that.
Some people might have.
Oh.
I was told that.
Maybe if you implement the task in some kind of video game that could like require the subject to memorize the image, that will improve the subject's engagement in the scanner. But it might be a bit tricky to set up, but since
there's already a video game branch in the program, so it might not be that hard to implement, as you say.
I know that, the idea is that even if the tasks become a bit more complicated than just regular memory tasks, you will still be able to use the data because you want to build like models on this neural data. You don't necessarily want to infer exactly which process is altered by the parameter you manipulate. So that might be an option that suits your purpose.
It could be. Thanks.
But I feel like it's hard here because we have no idea really what the task is. So maybe you could try to organize a breakout session next week to actually show the task when you have something working or just your computer.
Oh yeah, if I
And,
can plug mine,
so
it would be.
Build a little image, a dynamic image that just shows an example of two.
Or a GIF file.
Sorry?
Kinda like a GIF file.
Yeah, exactly.
Oh.
Like a file.
Or a GIF file that you can post on the Slack.
Maybe
Yeah.
So
I could.
But I really like the idea of trying to find a game. So the games we're working on, they're games from the mid-90s. Any video game console or arcade game we can pretty much deploy. So if you find one that has those exact components of like needing to remember images,
You just
we
replay
could just
the
put a game.
Super Mario Bros. 3 has a has a memory sub mini game inside that you get more lives when you memorize the cards. So
Exactly.
There we go.
Exactly what you need.
So maybe you could do a
Maybe we
little,
could
like,
give it a try.
literature review on on which classic game would have those components.
that would be a good plan. So it's not that much because I cannot show you my demo, but here it is. If you have any other questions.
Yeah. So what's the outcome of your project? What is it you want to
Oh.
produce exactly?
The outcome would be to have a data set and ideally the bits format, applying the FAIR principles to have it like distributed across the world so people can have it for free.
So these tasks.
These tasks, I'm basically trying to copy a puzzle without the box because I don't have the access to the data set. So I'm starting from scratch to try to reproduce it with people who have access, but I can't even download the original one, so it's protected somewhere on the internet.
But so what's
So-
the outcome exactly? A set of
The
images,
outcome-
a protocol to show stimuli?
It's a part of the Phantom project for Neuromod.
Yeah.
Oh yeah, I haven't even thought about it. The goal is to create a virtual kind of like brain with deep learning to eventually replace lab rats. Well, that would be in the future. Like I'm not sure I'm gonna see this alive, but virtual lesion studies and stuff like that. Compare Alzheimer versus control patients based on the activity of some people who are being constantly scanned every week for 5 years. So we get a lot of data to make a model, a solid model.
That's a general project, but your BrainHack project here, are you going to create PsychoPy script?
Oh yeah, I have that. It's just I cannot show it now, but up here this is this would be the task. It kind of works, but it's very buggy and it's not
And what
fully
kind of
reliable.
tools do you want to learn while creating that new or fixing, I guess, that script?
More Python, I guess. I'm still learning to make class-oriented programming. That's what Python is about, essentially, I guess. But
You can do object-oriented.
yeah, okay, yeah, that's what I
programming in
thought.
Python,
But
yeah. Yeah,
so
So, but you think you would be able to share a demonstration of that task at some point?
yeah, if I
Outside
could
of
just
the
plug
file?
my outside of that, how? No, I have no idea how, but I'm sure I can find it, or someone does know the answer, maybe.
Sorry, what was the question? I have
No, I thought you had a question.
no.
Anybody else? Right, thanks a lot.
Okay.
Well, hi everyone. My name is Nick. Just a quick acknowledgement to Pierre, to all the teachers. I'm having a great time, learning a lot. So thank you so much, and to all my colleagues. So I am entering my master's at University of Toronto and Rotman Research. The data that I've been working with is as a research assistant with Morris Moscovich at U of T. So it's basically, the data is a task-based fMRI, and it focuses on memory. So that was a nice leeway from Francois, memory to memory. So the study is, it presents images, if I can just show you quickly here. So on the left panel is study, and on the right panel is test. So participants are shown images of scenes at study, many different type of scenes. Here we see like a storefront, and then later on at tests, they're shown scenes that are either repeated, so either the same, or something here is called a lure, which is something that's similar to what they saw, but it's not exactly the same. And they're asked to give three responses, whether the scene they see is old, similar, or new. So this is a type of long-term memory test, and what we're trying to do, what we're interested in, is basically trying to look at the hippocampus, as some of you may know, that's involved with memory. We wanted to see that because the research has shown that in the hippocampus, neuroimaging data, fMRI neuroimaging data have shown that the hippocampus, along the hippocampus, the anterior portion of the hippocampus, like near the front, is associated with a very general, global, coarse recollection of details, and the posterior of the hippocampus is more associated with fine-grained details. So we predict that with this test, that the posterior hippocampus would be lit up if participants are able to correctly identify these similar looking lures, like these similar scenes, and the anterior would light up if they make a false alarm, like a miss. If they think that they saw something that they think that they saw the image before, but even though it's changed slightly, or it's a different type of storefront. So we're looking at the hippocampus, and the design here is using a T3 scanner at at U of T. We have basically it's 8 runs. It goes from study, test, and then a NAT scan, and then study, test. So there's 4 pairs. Each pair has different sets of scenes. They don't overlap, so participants won't complain too much. So the purpose with my analyses or my data set is that I'm hoping to use my DICOM data and either try and pre-process with fMRI prep. Alternatively, my lab has already pre-processed the data using SPM and MATLAB. So if along the way, because I'm using Windows and I've been having difficulty, if I don't manage to pre-process with fMRI prep, I can always use the already pre-processed data and then put it through NiLearn and try and look at some of the analyses of the bold signal of the ROI hippocampus or the whole brain activation, and then spit out the analyses or the visualization through Jupyter Notebook. So far I'm trying to use DICOM2BIDS, but Pierre has recommended me, instead of using DICOM2BIDS, there's another tool which is called, let me see, where did I put it? Convert? Right. Yeah, Hooticon, yeah. So hopefully that works. So yeah, and then that's my deliverables and to-do list. So far I'm a little behind, I'm embarrassed to say, but hopefully I can get started along. And then with Pia temporarily sponsoring me for the Compute Canada, I can try and run some analyses if the pre-processing do not work. Thank you. Any questions?
That's not a question, but I haven't seen Zaida, but you guys should work together because I know that she's preparing a big resource for everything that's fMRI and pre-processing and whatnot. So
okay.
You should get in touch.
Zaida unfortunately did not feel well and was not able to come this afternoon. I hope she's going to be able to present on Monday. You have to go.
So I'll see you next week, bright and early, 9 a.m. at Polytechnique, and looking forward to working with you. Sorry I have to leave a bit early.
Bye, Nicolas, thanks for coming. So Nicolas Sikov is organizing BrainHack next week. Yes, definitely, and I like your idea of advancing in parallel with the SPM preprocessed data if you cannot really preprocess here, because preprocessing data, even 30 subjects on a laptop, is a terrible idea.
Yeah. I was going to only do it for one person and
It just burn your machine. So I think Compute Canada would be really the way to go for the preprocessing. In terms of UDCON versus DICOM2BIDS, it's one of those, there's multiple sort of solution out there. I'd recommended to DICOM2BIDS because the main developer is located here, so you get the support. UDCON is also another one that is maybe a bit more powerful, but also maybe with a bit of a steeper learning curve. But they'll both do the job, so you can get a feel of the documentation and go with whatever you feel more natural.
Hope so.
So if you want to say sometime with the fMRI pre-processing, maybe if you don't intend to use pre-software or surfaces, you could skip that step.
Yeah, I could skip. Yeah, jump straight to NiLearn. Yeah.
Absolutely. What Basil was saying is that there's several options in fMRI prep, and doing the surface-based analysis.
Oh, okay.
Is way slower than volumetric based. So if you skip the surface extraction, you can save tons of time preprocessing your data.
Oh, okay. Thank you.
All right. And so maybe last question, that you said you wanted to do the activation estimation with NiLearn. Did I understand that correctly?
If there is, I haven't looked it up. If because it's task based, we have specific contrasts. If with the behavioral, so accuracy hits, misses, and see which area is associated with the contrast of.
So what you can do easily with NiLearn, well, easily I don't know, but it will be to try to predict based on the window of activity, maybe in the anterior hippocampus, whether people will remember or not the images correctly. Or maybe based on the activity of the brain while they're watching at the image, if they're going to say if they recognize it or not. That's more a brain decoding task. For brain activation, it's not really in there. There's another project which is called NiStats that does that, but it's very much of a work in progress. so it doesn't have all the polish and the testing of nylon. But the same team, so it's well done. There's nice tutorials and everything, but it's just not as developed. It's in alpha stage.
At least it's there, so it's good.
It's there.
Thank you.
Okay, thanks a lot. All right, so almost the end.
Second to last.
Then second to last, Norman. Just refresh.
All right, hi everyone. For those of you who don't know me, my name is Norman. I'll be starting my undergraduate studies at McGill in the fall in software engineering. So for a while now, I've been interested in machine learning. And so when I joined BrainHack, my main focus was to gain some practical experience training models. And in my opinion, one of the most interesting applications of machine learning is in the field of neuroscience. And so I decided I wanted to give a try at brain decoding, even if I had virtually no neuroscience background. So throughout this project, I'm hoping to familiarize myself with data science tools such as Nylearn and scikit-learn, as well as open science tools such as Jupyter Notebooks and Binder. So that brings me to my goal, and my goal is to create an accessible, extensive, but yet easy to understand fMRI brain decoding tutorial for those who are new to the subject, like I am right now. Pierre did bring up that maybe I could, once I finish, I can share this with the Nylearn team, and I would be honored if my project was good enough to be shared onto their website, but we'll see when we get there. So this tutorial that I'm planning on creating will differ from current tutorials, and that it will include the really truly basic fundamentals of fMRI analysis for those who have never worked with it before. Because the examples I've seen so far, although they contain clear and concise code, I find the underlying processes of fMRI analysis itself still remain obscure. For example, here's a brain decoding tutorial from Nylearn, or a snippet of an example. And although it is still well documented and the code is nice and concise, when I look at this, it says now we need to use the nifty masker. And I don't really know what a nifty masker is, given I don't work with brain data. So then I'll click on the nifty masker function, and that will bring me to this function signature. And the first sentence that I read is, applying a mask to extract time series from NII-MG-like objects. So I'm not really any closer to understanding what a mask is, but at least I did learn something. I learned that NII-MG-like objects are a thing, and I also need to learn what that is. So anyways, really what I'm hoping to do with this tutorial is to learn what these terms and concepts are, learn how to use them, and then summarize all this information in one place for anyone else who's like me in the future and who'd like to get their foot in neuroscience analysis. And to be clear, I'm not saying that the documentation is bad. I'm just confused because of my ignorance, and this project is a great way for me to start learning, really. So I don't know if any of you guys watch The Office, but this is how I feel right now. But hopefully by the end I will understand something. The main materials that I want to use will be Jupyter Notebooks. I think they're, as you all know, they're like intuitive. They're great for explaining concepts, and to increase accessibility, I'm hoping to make my repository binder compatible. So the main steps, first thing is to learn what the heck fMRI imaging is and how that data is stored. So I just got to read. I have to learn how fMRI data is analyzed, and for that I have to read. For my data set, I'm thinking of using Haxby's 2001 data set that investigated where it was a study where he investigated patterns of response evoked in ventral temporal cortex by faces and multiple categories of objects such as chairs, shoes, and I think scissors and stuff like that. And it's publicly available for me to analyze, so that should be fine. Some of the models I'm planning on using, I wanted to start a bit simpler, so I'm going to go with some of the basic things that were meant, like, it's the kind of the idea that was brought up during François Lespinas's presentation. So I wanted to try some basic models at first, and then if I have the time, I can go on to more fancy models. So I was thinking of just starting out with support vector machines, k-nearest neighbors, logistic regression, and then if I have time, I could try a neural network. And then finally, I'm gonna have to review my results, see it worked, see what didn't, and that's it. The pieces of my project, so this is what I'm planning on having. So a few different notebooks for the fMRI part, a little machine learning summary, the brain decoding tutorial itself, and then a discussion notebook just to finish it all, and then of course the requirements text file. And that's it. Any questions? Yeah.
I was wondering what kind of readings you had in mind, because I know there's a good pull track.
Yeah. So actually, 2 people recommended that book, and I am reading it currently. I'm also just really like searching the entire internet for any resources I can find that can help. And of course, anything that, or any of the resources I think are worth reading, I'll definitely include within my project. Yeah.
So maybe just a quick comment on the format of the tutorial you were discussing. So Nylern has documentation you have for each function, a detailed list of the arguments. So that's the second thing you were pulling out. The explanation about what the functions do are really, really short. Then you have like short tutorials, the first thing you were showing.
Right.
And there's a variety of these. So maybe in the tutorial on brain decoding, they don't explain what an NIMG-like file is. But if you go into the tutorial on loading fMRI data,
yeah.
they discuss it a little bit.
Mm-hmm.
But in any case, they're like short tutorials. And they're not necessarily meant to be for a lay audience. So I think what you're planning on doing does fit a need. And if you want to have an inspiration of the type of format, maybe the tone, the level of details you would like to get, I would recommend checking out the SciPy lecture notes.
Okay.
They've got some fairly detailed sort of like notebooks or just like pieces of code that are meant really as a companion to full-on workshops.
Okay, cool.
And those are typically much more readable and detailed. And I think that's kind of what.
Yeah, that's actually that's what I was thinking.
So actually, I said SciPy. I think it's like it's an sklearn like tutorial, but hosted as part of the SciPy lecture notes. Anyway, I'm a bit confused. I'll find the link.
You can open an issue and just propose it.
Yeah. Well done. Well done.
Props.
That was Rosé. Any other feedback for Norman? So, okay. Thanks a lot.
Thank you.
Okay, so we still have 15 long minutes for our last presentation, so we're right on time. I like that for Friday evening. And the last talk is for Jan.
Yeah.
So I hope you're going to be entertaining because people are like eager to go on weekend. So pressure's on.
So usually I spend too much time talking, So since it's the end of the day and I have plenty of time, so
you need to keep it short.
I will go through all the material I prepared for you. No, so my name is Yann Arel. Hi everyone. I'm a PhD student with Karim Jerbi at the Coco Lab. And what I will work on during this month is a side project. So my thesis focuses on sustained attention and neural networks dynamics during a sustained attention task, and during also various conditions of naturalistic viewing. But what I'm really interested into in my scientific heart is the relationships there is between the brain networks, the large-scale networks, and phenomenology and subjective experience. So I want to understand how various states of consciousness rely on neural network dynamics, and for that to explore a little bit this subject with Antoine, who is a good friend of mine. We started developing an artistic device which is based on neurofeedback, and the end goal of this device is to induce altered states of consciousness through the use of neurofeedback in an artistic and ludic manner. So this project was called the Coco Brain Channel, and it involves a pure data patch which generates music and visuals, and also Python scripts that handle like machine learning classification on the signal. And it was first developed to be used with the Muse headbands. And the thing is that Interaxon, the team, the company that developed Muse, recently decided to stop the support for their software development kit. So it was a sad news for us, but hopefully we found that there was a Python library that's called EEG Synth that could perfectly replace the proprietary software that we were using. So one of the main goals I had when entering this month was to switch from closed source softwares for this project to more open source solutions. But this isn't fun enough for a BrainHack project. This is like a very painful process of trying to understand how to redo what you did pretty well the first time. So let's not stop here. As I was saying, I'm interested into large-scale neural dynamics, and maybe some of you are familiar with this idea of self-organized criticality. It's based on the notion that spontaneous activity in the brain displays very, very complex patterns of activity, and that suggests that the brain operates at a point which is near criticality. And what that means is that small changes can lead in the brain. Small changes can lead to big differences. And the reason why it's interesting for the brain to work that way is because it allows the information sharing processes to be at the same time flexible but also stable. So information can be maintained and it can be also altered in an efficient way. So why am I telling you this? Well, it's because these ideas about the brain led us to consider the use of various measures that comes from complex system theory and apply them to brain activity. So there were like studies using DFA, Hurst exponent, or entropy to discriminate between different psychological states. And even if it's a literature that is pretty confused and nobody really knows what they are like what they are functionally measuring. They know the mathematical objects, but what they reflect is pretty vague, and it's even more vague for me because I don't have a formal background in mathematics. But I felt like using these measures in my neurofeedback application could be something very fun and could lead to interesting exploration of these complex features. So the end goal will be to use EEG synth to do real-time neurofeedback based on a Muse EEG headset. And more of that, I want to add a module in EEG synth. I want to start from an existing module that computes spectral features on the signal, and instead of computing the spectral features, I would like to compute complexity features. So that would require a wider time window. That would require some different stuff. But I intend to produce a module that is really easy enough to use, so it can be added to the existing library. So yeah, here is a small list of what I succeeded in doing for now. I can run EEG synth without any problem. Oh yeah, I didn't describe really how EEG synth works. It's a very complex modular library that you use by just connecting Python scripts together through ini files, and the base of this software is to send the EEG signal into a field trip buffer, and then to communicate the signal through a Redis database to all the other modules. So these modules will allow you to control your signal using a MIDI controller, for example, or even to do post-processing. So that's what I will be doing. I will need to grab the data from the Redis database, store them in a buffer, compute the feature, and then put back the feature in the Redis database to send it to Pure Data using OSC. So it's quite a complex pipeline. Yeah, what I can do now is to stream the signal to the field trip buffer and access it from the Redis database. I started creating the feature extraction module, but I still have some troubles to make everything work. For now, I obtain mainly nans, which is not the result I intend to get, but I will tinker on it, and I think that might go pretty well. And then after that, I just have to calibrate the data and send it back in via OSC. So my main objectives are to succeed at doing this, but I also have secondary objectives that are closer to the deliverables, which is, for example, getting a pull request for the complexity module I'm going to build. What I can also do if really I get bored and I finish this like really fast and everything goes smooth, and which is already not the case, by the way. What I could do also would be to build another module based on the same basis, but this time to re-implement the machine learning classification part of our setup, which could be a bit of work too. And also, this is something we talked about Antoine, and this is something that he would like to do because of his musical background. I don't understand a lot of stuff about this, but he wants me to create a module to perform peak extraction in real time. So you have the EEG spectrum, and then you can decide in the various frequency bands like the number of peaks you want to obtain, and that can be used also, the frequency of the peaks can be used as a feature for neurofeedback. And so yeah, for my deliverables, I intend to write a nice markdown file that will explain everything, every pain I went through in the process, and what was my start and where is my stop. I want to have a working version of the neurofeedback setup that maybe I could make a demo to you at the end of the month. You could even try it by yourself and tell me if it works as intended. And yeah, what would be really useful for that would be to end up with a nice Docker that could like easily install all of this, because it runs like it's based on Python, pure data with a lot of different libraries, EEG synth, which all have like different subtleties to get installed. And yeah, at the end of this, I could complement the markdown and the blog, like the blog post with a nice YouTube video that's easy to show what the experience is really about. So yeah, that's it.
Wow.
You see, I can talk a lot.
I see that. Any feedback?
I have a question or comment, I'm not sure.
Yeah.
It's a really, really cool project, and I was wondering, so maybe related to the complexity measures or the feature extraction, and how like a lot of the measures return like not
Yeah.
variables. Because I'm surprised because you're using a Muse headset, right?
Yeah.
It's only like here, right?
Yeah, exactly.
I wonder if it's like, is the amount of signal or like the amount of channels you have for sort of like such a complex sort of state analysis, is it enough to be able to extract a bunch of features?
That's a really good question. Like mid-term to long-term, we would love to get rid of the Muse headset and go towards like more like fuller coverage of the scalp and maybe more robust signals. But for now, yeah, we have 4 dry electrodes. So the first thing I can do to handle this problem is to average them together. That will reduce a little bit of the noise. But still, the signals are really noisy, that's true. And it could very well be that these complexity measures, when computed on these dry EEG signals, reflects the noise more than they reflect brain processes. So that could very well be the case. And since it's an artistic setup, I don't expect to have a way of detecting it if that was the case. Well, one way to detect it would be to try and just have a feeling of agency on the variations of the feature that you can induce. And if you are able to just let the neurofeedback setup run and control the feature, then it might be picking up something. But for now, since we don't really know what aspects of phenomenological experience are linked to what measure of complexity or so on, it's more exploratory, and the BCI approach we are using is more like a passive BCI, passive neurofeedback. So the features are just mapped into the sensorium, and you let the brain like do its predictive stuff like passively, you know. And so yeah, ultimately I would love to have benchmarks about this, but that won't be done with the Muse headbands and stuff like that. So.
Okay. Okay, so that's a wrap. We're still missing one presentation, hopefully early next week. So I hope you have a good idea now of the diversity, but also the convergence of some projects. Hopefully you can also start like collaborating next week. And I'm really looking forward to see what you are all collectively going to achieve, because it's a lot of stuff. And I wish you a great weekend, and looking forward to see you on Monday at Polytechnique.
Non, là ils sont à 9 maintenant.
Bye. Yeah, so what you're doing is very similar to what the type of data that François Pugam is analyzing. I don't know if it's necessarily this presentation was super clear, but the task is trying to encode these typograms. It's very similar to what you use. Show images, and so he's developed a database of about 9,000 images belonging to, I think, something like roughly 30 different categories, which can themselves be grouped into super-categories. So you have animate versus inanimate. Then in the animate you will have like the objects, and then the more like landscape or whatever. And in the animate you have the human, non-humans, and then the humans you have the faces, the body parts, and it's collected
Yeah, of course.
open images.
Yeah, we can see you. Yeah, we saw the video.
In, like consistent
Videos from London.
sort of format, and he's
Videos
gathered,
here.
almost 10,000 of those, and then he wants to create a memory task that will be a little bit like here you will have
How many has
What?
tested? Have you tested anything? He says we tested.
No, so the thing is there was a task that was deployed on this study. He didn't introduce that SIMAQ, which is an Alzheimer's study for elderly